Imagine this. You're riding your bike and doing a nice day, and then bam, you fall, scrape your knee, and now there's blood. It stings, it's messy, and you're thinking, great, now I'm going to have a scar forever. But wait, just a few days later, the wound starts to close up, new skin forms, and before you know it, you can't even tell you were injured at all. How does your body do that, you ask? The answer is mitosis. It's happening inside you right now, working like a tiny repair team, fixing injuries, helping you grow, and replacing old, worn-out cells. And the best part is, it never stops. Before we break down the steps of mitosis, let's talk about why it's so crucial. Number 1, growth. Every single person watching this started as just one tiny cell. Thanks to mitosis, that one cell kept dividing until you became the amazing person you are today. Number 2, healing and repair. Got a cut, broken bone, your body patches things up by making new cells through mitosis. Number 3, replacing old cells. Your skin, your blood, even the lining of your stomach, these cells don't last forever. Your body needs fresh ones constantly, and mitosis makes sure they're identical and fully functional. Without mitosis, life as we know it wouldn't exist. But what is mitosis? Mitosis is a type of cell division where one parent cell creates two identical daughter cells. These new cells carry the exact same genetic information, ensuring everything in your body keeps working smoothly. Think of it as like a photocopier. The original document, your cell's DNA, gets copied perfectly every time. No errors, no missing pages, just a perfect replica. Before a cell divides, it needs to grow, copies DNA, and it prepares itself, just like a student preparing for a big test. This entire process is called the cell cycle, and it happens in all living things from bacteria to humans. The cell cycle consists of two major stages, interphase and mitotic end phase. Interphase is when the cell grows, copies DNA, and gets ready for the division. It has 3 phases. During G1 phase, the cell grows and performs its normal function, like making proteins and energy. This is like a student collecting notes and reviewing the material before a test. And there's S phase. The cell duplicates its DNA, making an exact copy of all its genetic material. Think of it as like this, making a second copy of your test answers so you can share them correctly with the new cell. And then there's G2 phase. During G2 phase, the cell grows even more and checks for mistaken DNA replication. It's the final review before the big test. If errors are found, the cell fixes them to prevent problems in division. By the end of interphase, the cell has everything it needs: extra size, extra DNA, and enough energy to divide. Now it enters the second major phase, mitotic end phase. This is the phase where the cell actually divides into 2 identical daughter cells. The end phase consists of 2 main events, mitosis and cytokinesis. During the mitosis phase, the cell splits its copied DNA evenly between 2 nuclei. In the cytokinesis phase, the entire cell physically splits into 2 daughter cells. Now let's break down mitosis step by step. Mitosis happens in 5 key stages, and each stage plays a critical role in making sure that DNA is copied and distributed correctly. The first step of mitosis is prophase. This is where the cell starts preparing for division. What happens in the prophase is the chromatin, which is the loose form of DNA inside of the nucleus, condenses into visible chromosomes. Each chromosome consists of 2 identical sister chromatids held together at the centromere. The nuclear envelope, which surrounds and protects the DNA, begins to break down. Special structures called centrosomes move to opposite side of the cell. These help organize spinal fibers, which will later move to chromosomes. Prophase ensures that the DNA is neatly packed and ready to be separated without getting tangled. In metaphase, the chromosomes align up at the center of the cell in a straight line. Metaphase is like setting up dominoes in a perfect line. Everything has to be positioned correctly before the next step can happen. The spinal fibers pull the chromosomes, arranging them along an invisible line called the metaphase plate, which runs down in the middle of the cell. The cell performs a final checkpoint to ensure that each chromosome is attached to the correct spinal fiber. Now comes the action phase, anaphase. Anaphase ensures that each new cell will receive the exact same number of chromosomes. If this step goes wrong, one cell might end up with too many or too few chromosomes, leading to serious problems. The spinal fibers start pulling the sister chromatids apart, separating them into individual chromosomes. Each set of chromosomes is pulled to opposite sides of the cell. Finally, we reach telophase, the last stage of mitosis. Telophase is the final step in a nuclear division. It officially creates two separate nuclei, meaning the division is process almost complete. A nuclear envelope forms around each set of chromosomes, creating two separate nuclei. The chromosome loosen back into chromatin, returning to their relaxed state. The spinal fibers break down because they're no longer needed. Although mitosis is now complete, there is one final step before process is finished: cytokinesis. What happens in the cytokinesis? The cell physically divides into two new daughter cells. In animal cells, the cell pinches in the middle until it separates. In plant cells, a cell plate forms, which will later become a new cell wall. Without cytokinesis, we would just have one big cell with two nuclei. This step ensures that each nucleus gets its own fully functional cell. To observe mitosis in real life, we need a sample where cells are actively dividing. The best place to look? Garlic root tips. Since root tips are constantly growing, they have a high rate of cell division, making them perfect for studying mitosis. We'll go step by step through the process of preparing a slide and using compound microscope to analyze cell division. Before we begin, here's what we'll need. Now that we have everything ready, let's start with the first step, fixing the root tips. So we cut 1 centimeter of garlic root tips and place them in a fixative solution for 3 minutes to preserve the cells. Next, we transfer them into hydrochloric acid for 5 minutes to break down the cell walls, making it easier to stain and observe chromosomes. Then we remove the root tips, rinse them with distilled water and a few drops of 0.8% gentian violet stain. We let them sit for 3 minutes, allowing the stain to bind to the DNA for visibility under the microscope. Next, we place the stained root tip on a glass slide, add a drop of water, and carefully place a cover slip over it. We then gently squash the root tip with a razor and end of a pencil to spread the cells into a thin layer. We place the slide under a compound light microscope, starting at 10x magnification to locate the cells, then switching to 40x magnification for a clearer view of mitotic stages. Now we observe and identify the different phases of mitosis. Looking through the microscope, we see cells in different stages of mitosis. In interphase, the nucleus is visible, but chromosomes are not. In prophase, chromosomes condense and become visible. And in metaphase, we can see that chromosomes align at the center of the cell. During anaphase, we also can see chromosomes separate and move the opposite ends. Then we can see the telophase has two nuclei begin to form. Most cells will be in interphase, as this is the longest phase of the cell cycle.